Thank you very much. We'll get John to cut the arms off of that later on. Um, Thank you very much. It's been great coming here, and we'll just move forward. So, what we're going to talk about today is mitochondria, and don't have flashbacks to biochemistry 101. We're not going to go there. So, mitochondria represent they're in every cell of the body, and in the heart they represent 30% of the cell volume. So, the heart is really an obligate aerobic organ. Without the mitochondria, no energy would be there. It wouldn't work. And it requires a lot of oxygen to do this. And that oxygen goes through the electron transport chain and it produces the ATP and that powers your heart. So if you end up restricting that oxygen, you end up with what is major killer of the world, ischemia reperfusion injury. So this ends up with a lot of cell death. You end up with decreased cardiac function. And this is what this is gonna, okay, there we go. This is what a normal, I'm just gonna use a pointer. This is what a normal mitochondria looks like. It's on the M, it says M there, and it's electron dense. It's round and it's normal shaped. You can see them all aligned down within the sarcomeres on the side. That's a nucleus in the middle. And this is what they look like after just 20 minutes of ischemia. They get swollen, they change shape, they become translucent. They absorb a lot of calcium within them. And then you end up with decreased function. You end up with a decreased enzyme activity. You end up with DNA damage, and you change around the cell death pathways, get intrinsic, apoptotic, cell death, and you also change around mitochondrial transcription and proteomics. All of this occurs during ischemia, and even if you reperfuse the heart and reestablish the blood flow and the oxygen delivery to the heart, it persists into the reperfusion phase, and you end up with decreased cell viability and decreased function. Now, throughout the ages, there's been a lot of different methods to try to alleviate this. Most of them were either upstream of the mitochondria or downstream of the mitochondria. And for years, we had looked at this. I'm sure you've done it here. I've looked at it. We decided then around 2007 that why don't we look at organelle transplantation? Why don't we look at the mitochondria itself? Why don't we take mitochondria that are damaged and swollen? Why don't we take mitochondria from another place on the body that has not been affected by the ischemia reperfusion injury, take those mitochondria and then deliver them back into the heart to replace or augment those mitochondria that have been damaged. So we developed a procedure to do this. We take the tissue from usually the site, wherever we're going into. We can use sternocleidomastoid if we're doing a carotid cut down. Mainly, we started off using pectoralis major because we did a mini thoracotomy. And the babies, we always do a sternotomy, so we get the tissue from the rectus. But you can, if you do a femoral cut down, the vastus is right there. We take out 2 small pieces of tissue, number 6 biopsy punch. And this is an American dime, getting smaller as the days goes on. We just take those 2 small pieces of tissue, we put them into 5 mils of buffer. We do a mechanical homogenization, which nobody can, all you have to do is press a button and it goes. We add a little bit of a buff enzyme here to just dissociate the mitochondria from the endoplasmic reticulum. And this is the longest period of this isolation procedure. It takes 10 minutes here. And actually the 10 minutes, as I told somebody yesterday, you can work it around. That's a number I just pulled out of the air. So it's not like we went around, did 1 minute, 2 minutes, 3 minutes, up to 10 minutes. We just took 10 minutes. And at the end of that 10 minutes, we just passed them through a number of filters. And at the end, you end up with your mitochondria and you spin them down, you have a pure mitochondria. So this whole thing takes less than 30 minutes. Normal procedures take 2 hours. So this can be done clinically for the surgeons because no matter what you've heard, cardiac surgeons are not patient people. They're not going to sit around waiting for 2 hours while you isolate the mitochondria. So from those two pieces of tissue, you end up with about 1 times 10 to the 10th mitochondria. And they're all the proper size. Here's the size that they come out with. And they're all very pure. Here's an electron microscopy of the isolated mitochondria. You can see they're very pure, 99% pure. And the reason I know that is because one of the reviewers required us to do that. So you sit down a postdoc with a cup of coffee and a large bowl of anacin or whatever you want and let them count through countless electron microscopy. This just shows you that they're all viable. They maintain membrane potential. Here it is, one of Ibuki's facts showing you that there's 92%, I think, on that, or 92% is all viable. These are, the bottom is non-viable. And they maintain all of their electron transport and oxygen consumption capacity. This is important because mitochondria have to be intact, respiratory competent, and viable. If they're not, they won't work. So damaged mitochondria won't work. So this is just to show you what happens after you isolate them. This is just a CT. That is a stainless steel suture just to show you the location of the LAD. I've just put that in for identification. We've labeled the mitochondria here with F18 and rhodamine and iron oxide. And you can see if you inject them in by direct injection, they stay fairly local. Direct injection, we just use a tubercular syringe. Directly inject them in. You can, in an adult or in a baby, you can do 50 to 100 microliters. Direct injection right into the tissue. No need to do a purse string. The mitochondria are essentially homeboys. They want to get back into the cells as long as they're intact, respiratory competent, and viable. So you can put them in, they stay right where they are. Or you can deliver them through the coronary arteries. The way we do it is just do a carotid cut down, bring down a 4S or a 5S catheter, goes right down in the coronary ostium. There's no change in coronary patency, whatever. We deliver a 5 mil bolus, just like an adenosine bolus. So those of you who did in the cardiac surgery and have gone through with adenosine bolus, you know there's a difference between one quick bolus versus a long period of time period. A quick bolus is the best to go in. And you can see here that the mitochondria are only in the left ventricle. There's none here in the right ventricle whatsoever. So there's two ways of go through with this. And did I do something?
No, that's okay.
Okay, sorry. So you might think, well, if you're injecting a mitochondria and they're going to go all over the place. In fact, no. This is a 100 kilo pig that we've injected in 50 mils of mitochondria in one single bolus. It was a mistake. It was supposed to be 5 mils, but I handed it to my fellow and she got a little excited and shot all 50 mils in at one time. And even so, to my amazement, you can see that the mitochondria are only in the left ventricle. They're not located anywhere else. And here they are within the cells, and they go in very rapidly. And this is the same. You can do the same thing in the kidney. Here it is, just in through the renal arteries. We've done each one. The kidneys, this is a 100 kilo pig. Or you can do it into the lungs, either by direct injection through the pulmonary artery here. You can just see the lungs, or you can do an aerosol injection. And they go into the lung and the bronchi. And you can see they're in all the alveoli and the connective tissue. And here, the tubular epithelium, cortex, and the medulla. You can also do this in the skeletal muscle. It's localized, and it goes into all muscle types. So it's fast twitch, slow twitch, mitochondria are taken up all the way through. So how do they get in? Well, they get in by endocytosis. It's a billion-year-old process. If anybody remembers Lynn Margulis, she brought this out, talked about the endosymbiotic theory. Here we'd have just immunogold lib. Okay. Who's that guy? Okay. Here we have immunogold labeled mitochondria, and here you can see them at 5 minutes. Now we did this at 2.5 minutes, 5 minutes, and 10 minutes. I'm showing you at 5, but they go in at 2.5. It was the shortest time period we could do to stop the reaction. So at 2.5 minutes, you can see them adjacent to the cells. You can see them undergoing endocytosis, and here's the cardiac myocyte with all the mitochondria inside of them. And you can inhibit this by using cytochalasin D. It stops off actin-mediated endocytosis. There may be a little bit of macropenocytosis going on here, but it's the same thing, essentially engulfment, entrapment, the mitochondria go in. So, but as I said earlier on, mitochondrial membrane has to be intact. If you punch little holes in it with digitonin, it won't go in. Same thing with mitochondrial parts. If you take RNA, DNA, proteins, they do not go into the cell. So it's not like a bacterial uptake or a viral uptake. You have to have intact mitochondria to go in. The parts themselves will not go in. So once they go in, you can see them. Here they are labeled with a bacterial GFP. So that's stable transfection and labeling. And you can see these are going into a cell, cardiac myocyte, which has all red labeled RFP mitochondria. I'm going to stop that so you don't go into epileptic fits. Once they go in, you can see them on the early endosomes and then being released. You can see them on the late endosomes and being released. And you can see a small portion being associated with what looks like lysosomes. But anybody who's used those antibodies knows they may or may not be lysosomes. But they're released anyway, intact. Once they're released, they then fuse with the other mitochondria. You can see them here. They're actually fusing with the other mitochondria. See, here they're coming in, and there they're fusing with those other mitochondria. Once they fuse, you see all the normal fusion proteins, MFN1, MFN2, OPA1. We get a small, those are the fusion proteins. We get a small amount of DRP1, which is a fission protein, but it's not phosphorylated. So all the action that I'm going to show you is fusion, not fission. We don't have, the mitochondria are not decorated with Parkin or Parkin molecules. So we don't have that going on. So once they go in, you can see them. Here are the mitochondria that we go in. Here's the merge photograph. You can see them at 2 hours. You can see them at 1 day. Here it is at 28 days. They're still in there. And they're still functional. Here it is, normal total tissue ATP. Here's one with the mitochondria. Here's at 3 days, and here's at 20 days. So 28 days is when we finish. Essentially, our animal people do not want us in our animals in there after 28 days. We have to take them out, either bring them home or send them off to somewhere else. So we have done a lot of studies doing regional ischemia reperfusion. So what that is we just transiently snare the left anterior descending artery, tie that snare, do a temporary regional ischemia for 30 minutes. At the end of 30 minutes, we release the snare and reperfuse the heart. So that causes an area of approximately 30% of the left ventricle to be subjected to an ischemic insult. So we do that, and then during the reperfusion phase, we either give vehicle plus mitochondria or vehicle alone. So the vehicle is always the control on here. And then we reperfuse it either for 2 hours or for 28 days. And there's a number of studies that we've done here. Here's just a few of them. And what we continuously find is we end up with preserved cardiomyocyte viability. We get decreased apoptosis and necrosis. We decrease reactive oxygen species with the mitochondria. And we preserve the myocardial function and tissue ATP, and you get an increased enhanced MVO2 and ejection fraction. And there's a bunch of proteomics that I'll show you again here that are increased with this. And this occurs early. You can see these changes as early as 15 minutes. So as John had said earlier on, we and everybody else has been working with DCD hearts these days. The DCD offers a large population of viable tissue that can be transplanted to donors, to recipients. But the big thing with it is that you have this period of ischemia that is jeopardizing the heart at all times. And so how do you overcome this? Well, we decided, well, let's try the mitochondria on this. So we replicated the St. Vincent protocol. Sorry about that, but anyway, we used a 20 minutes warm ischemia and 10 minutes cold ischemia protocol. And we used all of our controls here that I'm going to show you are versus a DBD heart, which had no ischemia whatsoever. So we put in 10 minutes cardioprotective at the end. So it exactly matches what we had with the DCD. We started the warm ischemic time and when mean arterial pressure and central venous pressure was equivalent. So that was the start. And then 20 minutes warm ischemia. At the end, we did 10 minutes cardioprotective. And then we put machine perfused both hearts. Machine perfusion was 2 hours of unloaded heart, and then 2 hours of loaded heart. And this was always blood perfused in both of them. During the reperfusion phase, we did 2 protocols. We either gave just vehicle alone, and that was our control, or we gave vehicle plus mitochondria, and we did 2 doses, one right at the beginning and one just before the working heart. The reperfusion, we used the Manitoba protocol. It, is hypocalcemic, it's moderately acidotic perfusion. PCO2 was 45, PO2 200, and that's fairly high, but it's what we use for babies. So we wanted some of this work that I'll show you, we've done in both the adults, neonatal, and pediatric hearts. So we just kept it at what we're doing in our hospital. The perfusion pressure, 40 millimeters of mercury. Dopamine, we did a constant infusion of 4 mg per kg per hour throughout the perfusion. And we paced it when necessary, 800 to 100 beats per minute. The temperature, we just started off with 32, and then we gradually increased it to 35, 1 degree per every 10 minutes. So this is the system we used. It's not as advanced as what I saw downstairs with your ex vivo system, but it was nicely done. Usually you go into rooms where there's a blood perfused heart and you wear a raincoat and take in an umbrella, and the guys did a nice job here. This is all in, and it's a multi-mode system that is very cheap. As a matter of fact, it's just stuff you can get through the generally in the hospital. It's already been published and it's down there. The system essentially, here's the isovolumic mode where you just go through blood perfused through the aorta, and then you can switch over to the working mode. It goes through a compliance chamber, and what you do is most of it is through the aorta still, but you get some going down through the left atrium, and the pressures are 5 to 10 millimeters of mercury, and with a head pressure of 45 to 55 millimeters of mercury. So using that, these are just what we kept. This is the normals, what we have, the aortic root pressures here, coronary blood flow, hemoglobin, and atrial PO2 was all maintained throughout the 4 hours. Now, so this is what is normal. Here is the sham on the top, and is that pointer? Yeah. This is a sham on top. This is a DBD heart, and this is a DCD heart that's just had vehicle alone. Nobody's going to use that for any sort of transplantation whatsoever. But if you give them mitochondria, this is what it looks like. It brings back a DCD heart back to a normal. You get a little bit better. This is the serial mitochondria, and it's sort of what happened last night when we did an animal here. You know, the first shot it brings it back very well. Second shot brings it back even better. There's no statistical difference between these two, and I beat up my students quite a bit if nobody comes out saying, well, there might be a difference here, and I think you see, but there is no difference. These guys here need serial and single injection. There's no difference between the two. But if it was me being on the table, I would be putting my hand up saying, give me the serial injections, okay? So this is only an N of 6 to 8 in each group. So this is the same thing with LVPDP and DPD max. Here it is, the sham heart. Here's the vehicle heart, that DCD plus vehicle. But if you give them mitochondria, it's increased, comes back to normal. Same thing if we look at MVO2. Here's the MVO2 on the DCD vehicle, but if you give the mitochondria, it's actually an overshoot, and you get a lot better. This has all been published, and you're welcome to any of the slides anytime. So, no point in telling you what actually is doing, and it's better to see it. And the big thing about mitochondrial transplantation is we lucked out. We did the heart, and because it's instant gratification. Here is a DCD heart beating away. Here we are injecting in the mitochondria just into the aortic root, and this is what you get. And that just keeps going on for 6 hours last night, 8 hours some other time, or until you just say, stop. And this is what the infarct size looks like. Here's what you get in a DCD heart with no protection. And here's what you get with the serial or single or the serial injection. This is a control. And you can see that the DCD vehicle, here's a lot of contractile damage, a lot of infarct. There's a lot of edema here. But here it is a single or serial injection, brings it back, and here's a normal heart. So it acts very rapidly. Here's the mitochondria within the cells, just to show you where they are. So the mitochondrial transplantation does a couple of things. It preserves the mitochondrial cellular viability, and it decreases apoptosis and necrosis, and it preserves the function. So how does it do all of this? So we decided, let's look and see what are the pathways that are going on, the mechanisms. Let's look at the transcriptomics using RNA-seq, proteomics using SomaScan. You can use whatever you wish. We've used other procedures, and look at the metabolics. And let's see whether these results match what we're seeing functionally and biochemically on this. So what we found out is here's the RNA-seq data. Here's the DCD with the mitochondria, all these rows. This is the DCD with the vehicle, and here's the sham. And same thing over here with the SomaScan. Here's the DCD mitochondria in this lane, the vehicle, and the sham. And what you've done is you've normalized the transcriptomic and proteomic expression on the heat maps just by using the mitochondria. If you want to look at this on differential gene expression, if you look between the DCD hearts, they get mitochondria, and the DCD hearts, the vehicles, there's 826 genes different between those two, differentially expressed, and 142 proteins. And this is at a log greater than 1. So, you know, if you have, for old guys like me who've done a Western or a Northern, if you're looking at a blot and one versus another, this one is definitely dark. You can see it from the back of the room, even with glasses on, that there's a difference. So there's also the p-value is 0.05. So we've cut out a lot of small drug discovery on here, but what we do have is we know these are up and these are the ones that are changing around. Now, so that's the DCD mitochondria versus the vehicle. What happens if we compare the mitochondria with just the sham control? Essentially, there it is. It's the same. 9 genes are different and only 3 proteins. So essentially, we've reprogrammed the heart within this short time period. Once again, at that time. So what do we reprogram? Well, we've upregulated mitochondria, and you know, I can show you volcano plots and everything else. It's in the paper, but it doesn't really mean a heck of a lot. They look good, but essentially at the raw end point, here's the proteomic pathways. Upregulated mitochondrial pathways for oxidative phosphorylation, which we show, ATP binding, muscle development and muscle contraction, anti-apoptosis, which we showed you. I already showed you we get decreased cell death in there, removal of superoxide radicals, which we decrease the ROS, and RNA biogenesis. The RNA biogenesis would be for the transcription of the protein, translation of the proteins. And we downregulate fatty acid metabolism. If you get an ischemic heart, the first thing you do is move around what you're going to be using. And we downregulate proteolysis, which we've already seen. DNA damage is gone. Catabolic process, DNA damage, apoptosis is down. Intrinsic apoptotic signaling is down. The mitochondria are better. They're functioning better. And then we decrease the inflammatory and immune response. And all of this recurs rapidly. And if you look around, what's causing this? So both for the RNA-seq as shown here, and here by the SomaScan, mitochondria are a predicted source. So it's not like stem cells where you go in and it's a putative secondary messenger coming in. Here it is, it's autocrine action. It's not endocrine, it's autocrine. It's right directly related with the mitochondria. And once again, here it is with the metabolomics, and you can see that it's associated with the mitochondria and the skeletal muscle and the nucleus associated at that time. So the changes we're seeing physically with the mitochondrial transplantation are also evident both nuclear by transcriptomics, proteomics, and metabolomics, and all of them relate back to the mitochondria in that short time period. So we think the mechanism here is probably associated with retrograde signaling. Now, that would take a lot to maneuver around and prove and everything else, but all that means is rather going from the nucleus to the mitochondria or nucleus to the cell, we're now going from the mitochondria to the nucleus and then bringing it back to prepare the whole cell. So this may require you to look at things a little bit differently, turn it upside down a bit, but when you come back to it, it'll probably make sense. Now, I've shown you these changes. These changes only occurred at 4 hours. So the obvious question, well, does this persist? Well, here's a couple of papers that we've gone in a diabetic heart, and yes, they're up at 2 hours, and they're still there at 28 hours, 28 days, sorry. And they're up for anti-apoptosis, muscle development, muscle contraction, cardiac development. That's at 2 hours and at 28 days. And the downregulation, same thing, apoptosis, inflammation, DNA, and proteolysis. This is a model of diabetes. It's a standard model that everyone uses. And it was Zucker diabetic rat model, and we wanted to use that because we wanted a model in which the mitochondria were damaged, not by us during ischemia, but were damaged prior, and then we added mitochondria in there. Now, we obviously don't want to cure pigs and sheep, although it depends where you live and what side of the fence you're on. So anyway, I was originally at Harvard Institute of Medicine dealing with adults. And a good friend of mine, Dr. Pedro Del Nido Solution, many of you know that from cardioprotective, came over and they had a problem. They had some congenital kids congenital heart kids that were there. They had undergone some operations. They had a ischemia reperfusion, and this is a very small number. And then we had to put them on, they couldn't come off cardioplegia, so off the pump. So we had to put them onto ECMO. The problem with that is if you don't get them off ECMO within 72 hours, 89% have to go for redirected care. And we do not have VADs or heart transplants for the kids at this age. So this is actually one of our patients and the mother, and they've agreed to be in here, and they actually do very much. This is baby Avery at the time point. So under IRB, and we were allowed to treat these kids. And this is where John was coming in earlier on. In order to do this, you need cooperation. You need somebody who's a leader within your section is saying, we want to do this. And you need somebody at the hospital level who's going to say, yeah, the kids need this. Luckily, we had Pedro at the department head, and we had Sandy Fenwick, and she said she was the only CEO I've ever met. You know, I've been at a lot of hospitals throughout my career. I've never known a CEO. Never, well, today, two. Now I know two. So those two, that is what is required to move anything from research into the clinical phase. And if that's not there, it's not going to work. So anyway, we ended up with, I'm going to show you, the paper is on 10, we've done more since that. And we had a control group of 14. This is a historical control. You're not going to go into a patient and say, we're either going to give you a kid mitochondria and save them, or we're going to give them vehicle, and no, they're not going to be saved. So anyway, we had, all the patients had ischemia reperfusion injury. They were severe to moderate. As you know, with children, we do severe to moderate. We don't do a numbering, but I will show you numbers later on. So it goes for, severe to moderate is, for those of you in cardiac and cardiology, that's around a -6 to -8. The ECMO, they'd all been on ECMO for 3+ days, and they're unable to separate, and it was unlikely that they were going to separate spontaneously. So the mitochondria was delivered by, this is my colleague, Dr. Ram Amani, putting them in. You can see we just load them into a tuberculin syringe, and they're being injected into the heart. And we did 10 to 20 separate injections, 50 to 100 microliters each. Easily done, except on the posterior side. It's a little difficult with a syringe, but it can still be done. You can, normally what we do is we get a small piece of tissue. He gives it to me. We isolate the mitochondria right at the end of the bed. And I hand him back the syringe loaded with the mitochondria in 1 ml. And he just injects them in right away. And the injections were about 0.5 to 1 times 10 to the 7 mitochondria. The dose for the heart is around 2 times 10 to the 5th to 2 times 10 to the 6th per gram wet weight. So on an adult heart, that's about 400 grams. That's around 1 times 10 to the 9th mitochondria. So this is what it looks like. That's the heart prior to the mitochondria, and this is after 24 hours later. So it goes from -8 to -26. Most of us are sitting around here at a -20, -22. And it goes on like that. So right now we've done 17, and this is baby Avery at 2. She went through a full terrible twos, according to her mother, had no problem with that, didn't miss a beat. She's now 7, running around, having none of our patients have ever come back with cardiac difficulties. They've done 17. It's all decreased myocardial injury in all the treated patients. It's improved the functions within 4 to 6 days of giving them the mitochondria. We have an 80% successful rate. There's been no adverse experiences with any of them. No immune response, no autoimmune response, no inflammatory response. And the follow-up now is 8 years, and it's been very successful. So we have been focused primarily on the heart. It works in right heart failure. It extends the compensatory phase. Diabetes, cardiac failure due to diabetes, it works great. Here we're doing the work on the organ transplant, and we have an N of 1 from last night, but it worked out very well, and I think the guys are fairly pleased with it. This has been going on. People are using it in Sweden for liver, kidney, at the Karolinska and at Lund. Sandra Lindstedt and Gabriel Ionescu are using that. For the kidneys in the US, it's Dr. Orlando from Wake Forest. There's other people coming in for stroke. Dr. Becker is doing this for stroke in New York. In the eye, and also Melanie Walker at University of Washington. And now we've just been starting to do this in the eye for renal ischemia. So we think it has a lot of possibilities. It's simple, it's easy, it's quick, it's rapid. What I showed you here on the movie is what we got last night. For a cardiac surgeon, it's instant gratification. You don't have to wait around weeks, months, days to try and see what's going on. So with that, I want to say thank you very much. This has really been a team effort. I haven't got I left the slide of all the people working on this. Sorry, Ibuki, but she should have been here there. And with that, I'll say thank you very much.